Okay, and so they decided to try and figure out which part of the plant the sun was acting on to causing the bend to occur. So I'm going to draw a sun over here and a sun over here. So the first thing they did, they had a transparent cap. And when they did that, it bent towards the sun. Right? Still with this little cap on. And then when they had an opaque cap, it did not bend. So I'm going to draw arrows to show. All right, well, they thought, well, maybe it's the middle, the coleoptile, the shaft part. And so they put on a transparent sheath. And, of course, it bent. Okay? But why did they do this?
They wanted to see which part of the coleoptical is it called?
Mm-hmm.
They wanted to see, depending on which part they covered, if it would bend toward the sunlight.
Right. So why would they do a transparent sheath? Of course it's going to bend, right? So what's the point?
It's a control. It's a prevent sun.
It's a control. What is it controlling for? Well, the light is still getting through no matter what, right?
The effect of the sheath on the plant.
Right. So just the effect of the sheath. So maybe just the stimulus of the sheath, right?
Physical thing of it.
Exactly. Okay. So then they put a opaque sheath. And what happened then?
It didn't bend when it was on the. Oh wait, did it bend even when it was on like the middle part of the coleoptical? Yeah.
Yeah, so it's still bent.
In other words, it was definitely the tip.
Right. So the conclusion of this is the stimulus to the tip caused the bending, right? And we'll call this effect phototropism. Okay, which is bending towards or away light. Okay, and this was the same Charles Darwin who came up with the theory of evolution. Okay. So, and his son, so Frank and Chuck Darwin, played with some leaves and figured out, or plants. Okay. So along comes the next guy. Boysen Jensen. Okay, and he decided to cut the tips and then put this permeable gel between the tip and the coleoptile, the shaft part. And then he had an impermeable gel and did the same thing. Okay, expose them to sunlight. And the one with the permeable gel bent towards the sunlight. And the one with the impermeable gel stayed the same.
Dr. Hurt?
Yeah.
Can you explain a little bit more of how it was possible for, even though when the top of the coleoptical was cut off, that it was still able to receive nutrients from the sun and was still able to bend toward the sun? Just kind of.
Well, the gel was permeable, so he cut it off. I drew some separation there, but then he put it all back together like a sandwich.
Okay.
So whatever was going on in the tips could still go through this little gel down to the other part of the plant.
Okay.
But not on this other one. On this other one, it prevented it. Yeah.
So it's like a sandwich, like the top and then the gel.
Mhm. Yeah. So there wasn't a space here. It was more like this one on the right.
Like here's it's kind of like grafting in, so it's stuck together.
Right. So what's the conclusion based on these experiments? Talk to you guys too.
Something passes
Okay.
the top to the bottom that makes it bend.
Right. There's a signal from the top that goes to the bottom. Or to the shaft, causing it to bend.
Oh, really?
Okay? Everyone following this so far? Pretty simple?
Yep.
Okay, next guy. Went. Okay, so what he did is he took this permeable gel and he did all of these experiments in the dark, so without sunlight stimulus. Okay, and he took this gel and he cut off a tip and he put the tip on the gel and so basically allowed whatever was on the tip to seep into this permeable gel.
So the rest of the coleoptical wasn't below the tip of the other side of the gel?
Right, he's just using the tip. So he knows it's in the tip, that part doesn't matter. Just cuts off the tip, puts it on the permeable gel, and lets it sit there for a while to let whatever that signaling molecule go into the gel. And then he cut cubes, okay, of the gel and placed it onto a coleoptile that didn't have a tip in the dark. Okay? And it bent away from the gel.
He's a smart dude.
He's having fun.
Just like me.
So, what's the conclusion? Okay, the signal causing the bending. What else about the signal?
Physical opposite direction from the signal.
The signal is on the dark side. So it actually wasn't the light. I mean, the light actually does have something to do with it, but the darkness, the signal still occurred in the darkness, and it was the darkness which was causing the signal to be made and passed. And he then went and named this hormone auxin. All right, so auxin this is the first plant hormone that was discovered, and it has a number of different effects, but one of them is it helps plants bend towards the light. Okay. All right, how does it work? We're going to talk about it now. So here's a little cross section of a plant. We have our cell wall here. We have a vacuole here. We have cellulose fibers, and we're going to zoom in here. Maybe I should use a different color. These are cross bridges. Okay, so these cross bridges tie the cellulose strands together so that they don't move. Okay, so what happens is, first we have auxin that shows up. This is our auxin.
Oh, that's huge.
It is. It's ginormous. Okay, and what auxin does is it causes our proton pump to be activated. And we all know a proton pump requires ATP. All right, so the auxin activates the proton pump, and it starts pumping protons into our cell wall. So then you get a bunch of protons in here. And if you have an increase in protons or an increase in hydrogen ions, what happens to the pH? All right, pH lowers as a result of this. Okay, which means it becomes more acidic. Okay, this is the signal for another enzyme called expansin. To cut these cross bridges. Okay, but now that those cross bridges are cut, the pressure due to solute pressure causes then the cell to grow.
I'm sorry, can you zoom in a little bit on cellulose again? Okay, thanks, I got it.
Okay, the cell's natural state is that the internal pressure due to solute pressure is pushing against the cell wall, and the rigid structure of the cell wall is pushing back, and they're balanced, so it's tight. But when you release the pressure from the cross bridges, now the solute pressure from the inside of the cell pushes on it, and then it elongates the cell. Okay, you do this on all the cells on one side, and they will get longer, and it will cause the plant to bend. Okay, so this is how auxin at the cellular level then affects the bending of the plant. Yes?
So pH triggers the release of expansin, right?
Mm-hmm. Yes. Okay, so if we were to number these, okay, the first step would be auxin. The second one would be the protons going through the proton pump. And then the third one would be the pH decreasing. And then expansin cuts the cross bridges. And then the cell wall expands. Yes.
So just really quick, the reason why it expands is because the cellulose fibers like elongate, is that what
Well, the cross bridges are holding them in place. There's already being pressure because of the vacuole is holding all this water. So if you cut those cross bridges, now that pressure causes it to expand and they slide. There's nothing stopping them. Yeah.
So is that happening in the right, like in the bending side or in the opposite side?
On the long side, right? So
Oh,
it gets longer and it causes it to bend over. Yeah. Yes.
Why would it do this, though?
Well, it does this in response to sunlight, right? On the dark side, it has lots of oxygen, and so then it grows so that it can go towards the sun so it can photosynthesize more.
And then once it's done, will it retract back?
Yeah, so when the sun goes down, then it should just move back.
And then it re-engages the cellulose bridges, whatever they're called.
Yeah, so it's temporary. The bridges can go back after expansion goes away. Once this oxygen goes away, okay, then those protons can diffuse back in. Expansion will be deactivated. The cross bridges will reform, and it goes all in reverse. Yes. Yeah.